We are, so good afternoon and welcome to our third Signs of the Seasons webinar in our series this year. Through these webinar presentations, we've explored rockweed, loons, and now historical data in Maine. We're pleased to have Caitlin with us to share her exciting work with us. Signs of the Seasons is now in its 7th season, and we've had a great season thus far. I wanted to share a little bit of new information about us. We've just completed our 2017 Signs of the Seasons survey, which we've been doing every year since 2011, and had 91 observers participate in this. That's a record. So it's really helpful getting this information so we can work towards improving our program. These webinars, in fact, are a result of observers asking for more information on our indicator species and phenology stories. So let's get started. I'd like to introduce Caitlin Mcdonough MacKenzie, an ecologist and alpine zone enthusiast. She studies plant ecology above tree line in New England with an emphasis on understanding changes in plants' communities over multiple timescales, from the recent Anthropocene to the last 15,000 years. Caitlin studied environmental science and public policy as an undergraduate at Harvard and ecological planning as a master's student at the University of Vermont. Her master's project partnered with the Appalachian Mountain Club to study alpine plant phenology through the citizen science project Mountain Watch. So that's another partner of ours too. Her PhD in biology at Boston University explores patterns of species loss and spring phenology in New England's plant communities, and she conducted field work at Acadia National Park. She defended her PhD in May 2017 and will begin a postdoc as a David A. Smith Conservation Biology Fellow at the University of Maine Orono this fall. And as a postdoc, she'll be studying paleoecology in Maine, looking at changes in plant communities above tree line through pollen captured in lake sediments in Acadia National Park and Baxter State Park. We're really happy that she's remaining in Maine and excited for her talk today. So I'm going to turn over the presentation to Caitlin. And that just takes us a few seconds. Thank you for being with us today, Caitlin. Really appreciate it.
Thank you so much for that lovely introduction. Can you see the screen now?
Yes.
Great.
Thanks.
So I'm excited to share my research with everyone about uncovering the past through Maine's historic phenology. And when I talk about that, hold on, I'm just gonna come out for a second so I can move GoToMeeting and see my full screen a little bit better. There we go. All right, slides look good for everyone. So I am really excited to talk about my dissertation research, and as Esperanza said, I defended in May, and this presentation, this webinar, is a little bit like the VH1 behind the music version of that defense talk, where I get to dig into the more human stories behind the data that I worked with. So I'm really excited to share this with you because my PhD would not have been possible at all without an amateur college botanist who was working on Mount Desert Island in the 1880s, or a school teacher who grew up in Concord, Massachusetts in the 1840s and 50s and summered on Mount Desert Island in the 1880s and 1890s, or a mid-20th century hunting guide who lived way up in Aroostook County. And we basically don't know anything about him except for what he left behind in daily journals. And then there's Henry David Thoreau, and a lot of ecologists and natural science folks like to point back to Henry David Thoreau as an influence on their research and on their enthusiasm about nature. But Thoreau had a slightly more direct influence on my dissertation. I worked in the Pre-Mac lab at Boston University, and the Pre-Mac lab has been using Henry David Thoreau's journals as a data set for the past 10 plus years. And in addition to that influence, the school teacher who grew up in Concord, Massachusetts, actually learned to botanize with Henry David Thoreau as her mentor. And she went on to mentor the amateur college botanist who collected all of this data on Mount Desert Island. So Henry David Thoreau really had an outsized influence on this dissertation. And I think it's only fitting that he was such a fan of Maine from Massachusetts, and I also grew up in Massachusetts and have become a huge fan of Maine. And finally, there's another person or fictional person involved in influencing my dissertation, and that's Samantha Parkington. And she is an American Girl doll. So for those of you not familiar with the American Girl doll series, this is a group of very beautifully made dolls that in the early 1990s were super popular. I think they're still pretty popular. But each doll came from a different historic time period, and there was a series of books that went along with them. So my grandmother gave me Samantha Parkington, who grew up in Victorian New York, and so she's about 10 years old in 1904 when her books take place. And so Samantha is grappling with things like industrialization and women's suffrage movement, and she has a very wealthy grandmother who takes her summering to a location called Piney Point, which could really be anywhere on the Maine coast. And in her sixth book, Samantha Saves the Day, Samantha is at Piney Point and she's got this cute little sailor dress, but she also has these really beautiful accessories, including a wicker backpack with a little butterfly net and some little tiny fake butterflies. There's a watercolor sketchbook and a really teeny tiny little watercolor and watercolor brush that you could actually paint inside of, and a little pine pillow that Samantha had sewn with pine needles from Piney Point. And my favorite little accessory that she had was this little tiny plant press. It was about the size of a Post-it note pack, and you could actually use it to press little tiny plants. So I would do this. I would go out into my yard and take little violets and cut them so it was just the top of the flower petals, and put it into the plant press and press plants with the Samantha Parkington plant press. And so Samantha provided for me this weird outlet of combining history and ecology and kind of understanding how botanists went through the world before I really had an understanding of what botany was. And so when I think back on the late 19th century and early 20th century, and the folks who were doing some of the botanical work that I now depend on, I always have this image of the context of Samantha Parkington and my tiny plant press. And so I keep Samantha there as my childhood reminder of I actually did grow up to be what I was pretending to be when I was about 10 years old. And so to just orient ourselves to what we're going to talk about today, I have Concord, Massachusetts, and that's a pretty big star for the lab that I work in at Boston University. We do a lot of work in Concord, and I'll continue to come back to Concord as a point of comparison with Mount Desert Island, Maine, where I've been working in Acadia National Park, and also for Oxbow, Maine, where I have the Hunting Guides Journal way up in Aroostook County. And so you can see this spans a pretty large area in New England. Excuse me. And we'll be talking about plant communities across all of New England and northern New England especially. And so I know this is a Signs of the Seasons group, and everyone is probably pretty familiar with phenology, but I wanted to put out a technical definition. When we talk about phenology, we're talking about the timing of seasonal biological events. As a plant ecologist, I'm often thinking about plant phenology, and I'm specifically interested in spring plant phenology. So I look at leaf out timing and the timing of flowering. And looking back over the last 130 years, this shows how the climate globally has changed since some of these early naturalists were collecting and recording and noting phenology. And thinking about the context of when all of these observations were taken, we can see that the northeastern United States has become a much warmer area compared to the 20th century average over that last century and a couple of decades. And so when we think of plant phenology as providing some of these indicators of how the climate has changed, we can think of things like the length of the growing season. And so when we compare the length of the growing season in Maine today to Samantha Parkington's time, to the time of the hunting guide in northern Maine, or to the time of the school teacher in Mount Desert Island, we can see that the growing season is almost 2 weeks longer than it used to be. And that's a really simple metric that's kind of easy to wrap your head around. But ecologists have been studying the ecological impacts of climate change for a couple decades now. And this has a huge range of possibilities. So climate change can impact the behavior of organisms. So in this example, the purple spiders have actually shifted down into the territory of the pink spiders as the top layer of the grass canopy warms up, and that provides a lot more pressure on the grasshoppers at the base of the grasses, but actually provides kind of a haven at the top of the grasses. Climate change has impacted the distribution of organisms and ecosystems. In Vermont, there's been a lot of work looking at the ecotone, or the boundary between northern hardwood species and high elevation boreal forests. So the northern hardwood species like maples and beech trees have actually shifted up in elevation and are found at higher elevations than they were 80 years ago. So areas that used to be strictly spruce and fir now have more hardwoods. Climate change is also impacting the abundance of organisms. In the high Arctic, Tape and his colleagues have noticed a shrubification of the landscape. So areas that used to be very herbaceous have a lot more woody shrubs, and they've expanded their abundance. And finally, phenology. Phenology is a great marker of the ecological impacts of climate change. And I love this example from Japan, where they've been tracking the fruiting phenology of cherry trees across an elevation gradient. And at the lowest elevation, the cherries ripen early in the season, and the bears eat those cherries, and then they start moving upslope to follow the ripening cherries. So as they move upslope, they're also helping to spread some seeds by depositing the seeds that are in their feces at higher elevations. So when we think about how plants and animals will have to track climate change by perhaps migrating upslope, the bears are actually providing a way for the cherries to do that, so that the low elevation cherry populations will be able to move up to the mid elevation, which will soon feel like a low elevation site, because the bears have already moved the seeds for them. And so that's a great example of phenology across different levels in the ecosystem between animals and plants. But not all of the phenological research that's done shows this great harmony between animals and plants, or different trophic levels. And so Linda Mapes wrote really beautifully that there are two calendars now. The seasonal timing evolved within living things, and the seasons cooked up by us. And I think when we study phenology as an indicator of climate change, we have to keep in mind that we need both of those calendars. We need to know what's happening now, but we also need that past calendar to provide context. So we need some historical data to understand how things have changed and what the shift has really been. And luckily, lots of natural historians have collected or recorded phenology data without any idea about the impacts of climate change. And so Henry David Thoreau is a perfect example, where in the 1850s, he was constantly recording the first flower, the first leaf out, migratory bird arrivals in spring in his diaries. And we in the Pre-Mac lab now read those diaries as if they are data sets. So we're able to look at how the phenology has changed since Thoreau's time in Concord, Massachusetts. And so this figure comes out of the lab. This is a paper that Dr. Pre-Mac worked on with some other grad students. And on the x-axis, there's the mean spring temperature from cooler years to warmer years. And the y-axis is the mean first flowering date from mid-April through mid-May. There's a really clear pattern where in the warmer springs, the first flowering date is earlier. And that's especially true in 2012 and 2010, which were extremely early years. So when we go back to Thoreau's Concord and monitor the same plants that Thoreau was watching, we can see how with warming temperatures, the spring has shifted. And there's a really nice correlation between the two, and we can actually describe quantitatively that for each degree Celsius of warming, the mean first flowering date is advancing by 3.2 days. So it's negative because that shift is happening earlier. And in this case, each of those data points represents 32 common plant species in a single year. So that one data point for 2010 is all of the plants in 2010, their mean first flowering date. And while that's great for a community-wide number, it's important to remember there's a lot of species level variation in phenological responses. So in this figure, which is also from another grad student in our lab, the x-axis has a whole list of different plant species. There's trees and shrubs and vines, and you don't need to worry about decoding what each of those abbreviations mean, but just that each of those is a different species. And the y-axis is the thermal units to leaf out in the field, which is just a jargony way of saying how hot before the leaf out occurs in the spring. And you can see that each species has a different requirement or a different heat level before it leaves out. And this pattern of species level variation plays out in studies across the globe, looking at leaf out or flowering or migratory bird arrivals. Some birds are more sensitive to warm spring temperatures. Some flowers are more sensitive to warm spring temperatures, and we see that over and over again. And that species level variation is important ecologically because it leads to lots of different effects throughout the ecosystem, from trophic interactions, where a hummingbird might depend on a certain flower's nectar. And so if that flower is blooming earlier and earlier, and the hummingbird is arriving later and later in the spring, there's a risk that they might eventually miss each other. To mutualisms like plant pollinator interactions. So the timing of when pollinators are in flight versus the timing of when these species are in bloom. Can also think about leaf out and carbon uptake. So when leaves first come out, plants will start photosynthesizing and pulling carbon dioxide out of the atmosphere. And that has huge implications for our carbon budget, or how much of a carbon sink a forest might be. And then finally, there are implications for competition. So in our lab, we found that non-native species like invasive Japanese barberry leaf out much earlier than the native tree species, and they're much more responsive to warm spring temperatures, which might allow them to outcompete some native plants because they're able to start their growing season very early and perhaps even shade out their competitors. And so all of these ecological impacts are important for conservation. And my dissertation research at Acadia National Park was a part of the climate change vulnerability assessment. And we know from other studies that plants that don't shift the timing of their phenology, so plants that don't flower earlier in warmer years or don't leaf out earlier in warmer years, those plants are more likely to disappear or decline in abundance. And so there's some kind of relationship between the fitness and the phenological sensitivity. And scientists are still working on that. I'm still doing some studies on that. But we know that that connection exists, so we can use the phenological sensitivity as a metric, and we can identify some plant species that are less sensitive, and perhaps those need more monitoring, or they need more habitat restoration, or they might need more help in terms of assisted migration. So using phenology as a way to understand which parts of a plant community might be most vulnerable. And so that's where my research really jumps off from. And I'll start introducing you to some of my collaborators. So this is a photo from the 1880s. And this is a group of Harvard undergraduates who had decided to come up to Mount Desert Island to do some research on their summer vacation. And Samuel Eliot and Charles Eliot, who are in the middle, were the sons of the current president of Harvard. And they hosted a gathering in the spring of 1880 for their friends. They invited their friends over to their dorm room, and they sat around and thought to themselves, why not select some particular region and put in the summer studying its geological formations, its flora and fauna, its birds and fishes, its trees and shrubs? There would be a happy combination of work and play, sea and land, tramping, sailing, and reading. And so they did that. They named themselves the Champlain Society, and they sailed from Boston up to Mount Desert Island, and they set up camp. This is one of their tents at the campsite, and spent their summer exploring. And they divided their society up so that one person would be the geologist, and he would study all of the rocks, and another person would be the ornithologist, and he would study all of the birds on the island. And one person, Edward Rand, would be the botanist, and he would study all of the plants on the island. And they returned for many summers to do this. So here's Edward Rand as a slightly older gentleman. In the second photo, he's in the back somewhere in the tent working on plants. So Edward Rand was among the most serious and studious of the Champlain Society members. And while the ornithologist and the geologist went off to the hotels in Bar Harbor and Southwest Harbor and socialized and perhaps got some girlfriends, Edward Rand remained very dedicated to his botanical mission. And he completed 14 years of fieldwork. So he returned to Mount Desert Island throughout his undergraduate career. He went to Harvard Law School and he kept coming back to Mount Desert Island while he was a law student, and then while he was a lawyer in Boston. And his 14 years of fieldwork were really impressive. And in fact, the second picture that has me and my daughter in it is in an exhibit that the Mount Desert Island Historical Society put on last summer, where they created the tent of the campsite and did a beautiful exhibit that honored the Champlain Society and the work they had done in looking at the science of the island. And as I said, Edward Rand was a lawyer. He was not a trained botanist, although he took college classes, but he was not a professional botanist at all. And yet he produced a huge amount of historical records that are just amazing. So he collected a lot of herbarium specimens. He used the same kinds of plant presses that my Samantha Parkington doll had. And the Champlain Society kept a daily camp log, so we know exactly what kinds of work he was doing, where he was going, what mountains he was climbing, his thoughts on collecting plants, as well as some of the fun antics that the Champlain Society got into together. And he really did reach out to the community beyond the Champlain Society to try to get other people involved in his mission to catalog the plants on the island. So the older gentleman in the picture who's actually making another pressed plant is John Redfield, who was a retired businessman and botanist from Philadelphia who lived up on the island. And then the woman who looks very stern is Annie Sawyer Downs, who is the school teacher from Concord, who came up to the island. And she and her husband built a house in Southwest Harbor that's still standing. And Annie Sawyer Downs and John Redfield collaborated with Edward Rand and mentored him. They were both much older than him. And they helped put together this catalog. And so their herbarium specimens are still at the Harvard Herbaria, and they provided a lot of help on the flora of Mount Desert Island, Maine, which is this flora that Edward Rand eventually published in 1894, 14 years after he had begun his work as a member of the Champlain Society. And we can use his historic data to look at floristic change. So every single species in that flora of Mount Desert Island from 1894 has a small description of its abundance on the island. And very fortuitously for me, I started my work as a PhD student right after the publication of the Plants of Acadia National Park, in which Glenn Mittelhauser and his co-authors had basically surveyed all of Acadia National Park in the same manner that Edward Rand had a century before them. And so Glenn Mittelhauser's book categorized each plant as being common, occasional, uncommon, or rare. And we worked with him to limit or restrict our analysis just to the plants on Mount Desert Island, so to cover the same geographic area that Edward Rand had been working on. And then I went through all of Edward Rand's lovely Victorian flowery descriptions of abundance and put them into one of the 4 categories that was used by Glenn Mittelhauser, so we could see how each species had changed over time. And our results look like this. So floristic change on Mount Desert Island from 1894 to 2010. The historic flora had 730 species. The current flora is 829 species, and 205 of those are new arrivals. There's been a net loss of native species and an increasing proportion of non-natives. 106 species that were recorded by Edward Rand are now locally extirpated, so they're no longer on the island. And that's just under 16% of the entire historic flora. Another 34.4% of the historic flora has declined in abundance. All habitat categories across the island have lost species evenly. So that's when we looked at things by if they grew in woods or grasslands on the coast, mountains, wetlands, or roadsides and disturbed places. They've all lost species. And three families, the lilies, the mints, and the orchids, have experienced very high species loss. And so to put that in context, I looked at other places in New England where there had been a published floristic change study. So this figure just shows what kind of historic data is available across these studies in New England. And you can see very clearly that Mount Desert Island is the farthest north of these studies and the only one in Maine. So that right there is just an important piece of context. But then on top of that, we have all sorts of different historic data that have been used. So for three of them, 3 Mile Island in New Hampshire, the Harvard Forest out in western Massachusetts, and Middlesex Fells just north of Boston, those are all based on published floras, just like the 1894 flora that Edward Rand published. The purple dots, which include Mount Desert Island, are places where we have a published flora and herbarium records. So we have the flora itself, but we can also go into the herbarium and see the flowers that were pressed by the folks who are actually doing that original survey. So we can collaborate, or we can say that their identifications were correct. We can see where things have changed names over the past century or so, or if scientists have decided that a subspecies is actually a species now, we can actually trace that on the herbarium specimens themselves. So it's like another quality control for assessing how well that historic flora was cataloged. And in addition to the herbarium specimens, on Mount Desert Island, we also have those camp logs. So we know exactly what the field work was like and how much effort they put into it and where Edward Rand went. So we have this additional level of information about the historic flora. For two of the historic floras, including Mount Holyoke Range, just north of Springfield, and Worcester County, we see that they just have herbarium specimens. And so that can be problematic because for example, in the Mount Holyoke Range, they were just looking at any herbarium specimen that had been collected during a historic time period. So there were multiple people collecting and they had different goals, and no one had this kind of central idea of, I'm going to go out and catalog every species that's here. And in fact, there can be a lot of biases in these historic herbarium collections. People often try to collect the prettiest things or the flashiest or most rare things. There's often a bias towards places that are very accessible, like along a roadside, or very inaccessible, like on top of a mountain, because there's more glory involved in that. But the kind of mid-range, relatively medium difficulty places to get to, or relatively not showy flowers, can be underrepresented. So there can be some problems with using simple herbarium records as a historic source. Let's see, Broadmoor Brook and Concord have unpublished floras, so as if the manuscript for the 1894 flora never made it out into the world. And there can be some problems with those. For example, they don't get feedback from the broader community. After the 1894 flora for Mount Desert Island was published, Edward Rand received lots of letters and accolades, and I actually went into the archives at Harvard and read all of his correspondence, and it was actually a really exciting experience to see how positively his work was received by the very new professional botanical societies around the country. Because again, he was not a professional botanist, so it was really nice to see these professional botanists writing lovely letters to him. And he also continued his fieldwork after he published his book, and then would continually update. So he would publish in the New England Botanical Club's journal updates throughout the 19-teens and 1920s of species that he had found or other people had alerted him to that had been missing from the original flora. So he had this very completist idea to it, which is missing in unpublished flora. And then our last example from Needham had an unpublished flora and an herbarium, so we can kind of collaborate or corroborate between the two of those. Again, just to put into context that Mount Desert Island is a really unique historic flora where we have so much information about who was doing the research, what they actually saw, all of the herbarium specimens, and then that published flora. And when we look at the rates of species loss across these floristic change studies in New England, we can see a huge range from under 10% of the flora, of the historic flora disappearing from a landscape to over 40%. And what I find interesting in this example is that Mount Desert Island is not the least species loss, or the place where the species are most protected, I would say. So Mount Desert Island has been a national park for 100 years, but despite this protection from development pressures, it's still losing species, similar to places like Harvard Forest in Worcester County. Worcester County does have some very rural areas, but it's also a highly developed area. And in each of these individual studies, the authors are often citing that development pressures are probably driving species loss. But when we look at it as a regional trend, it's clear that places that have different levels of conservation, from state parks like the Mount Holyoke Range to conservation easements like Middlesex Fells to national parks like Acadia National Park, they're all experiencing species loss. And these same families, the orchids, the lilies, and the mints, are losing species across the range. So that shows that there might be a more regional trend or even global trend in terms of climate change that's driving some of these patterns of species loss. So from that very big regional picture, I wanted to zoom in on a species level look. And so as I talked about before, our lab has done a lot of work in Concord, Massachusetts. So on top, there's a picture of my advisor and Abe Miller-Rushing, the science director at Acadia National Park, with Henry David Thoreau's statue at Walden Pond. I wanted to recreate that photo myself, but nobody made a statue of Edward Rand, or nobody has yet. There is, however, a little plaque to him on a rock in Acadia National Park. So that's me and Edward Rand's rock. And where Richard and Abe looked at changes in the flora of Thoreau's Concord to look at changes, excuse me, in plant abundance since the 1850s in Concord, I noticed that Thoreau's Concord and Edward Rand's Mount Desert Island had a lot of species in common. In fact, there are 304 species that are in both locations. So if we just zoomed in on those 304 species, I expected that abundance change in Concord would help us predict change on Mount Desert Island. So in my hypothesis, I have this expected idea that for all of the species that are declining in abundance in Concord, most of those would also be declining in abundance on Mount Desert Island in that first green bar. Or in that middle category, if they were experiencing no change in abundance in Concord over the last century or so, they also wouldn't have changed in abundance on Mount Desert Island. So that was my hypothesis. And I analyzed their abundance change in both locations for all 300 species, and found no correlation between what had happened in Concord and what we had seen on Mount Desert Island. So my hypothesis was completely wrong. At the species level, a species can be declining in abundance in Concord, and that same species could have increased in abundance on Mount Desert Island, or vice versa. So when we look at these floristic change patterns, we have this community-wide species loss that's similar to regional trends across New England. But at the species level, floristic change in Concord is not predictive of floristic change on Mount Desert Island. So it's really pointing to the fact that you need local data to make local decisions for management, especially if you're in a place like Mount Desert Island or in Maine, where there aren't a lot of local examples yet. All right, so let's move up to Aroostook County and talk about Quackenbush. L.S. Quackenbush was a hunting guide in the 1940s and 50s who lived up in Oxbow, Maine, just south of Presque Isle. We have no photographs of him. In fact, we know almost nothing about him besides what's in his journal, which includes daily records of temperature and observations of things like nature. He saw a fox. He has observations of migratory bird arrivals, first flower, first leaf out in the spring. And this data set has a really interesting backstory. So the Turrets is this cottage or mansion that was built in Bar Harbor on Mount Desert Island in the 1890s. So in fact, this photograph is from the same era when Edward Rand had just published The Flora of Mount Desert Island, and Samantha Parkington was about to go up to Piney Point. And the Turrets is now a part of College of the Atlantic. And a few years ago, they were doing renovations on this beautiful historic building. And during the renovations, they had to empty out the attics of the Turrets. And during that process, they found this box, which was just the stacks and stacks of the journals from L.S. Quackenbush. And the backstory was that sometime after Quackenbush died, and his last journal entries are from 1959, so supposedly sometime in the 60s, Quackenbush's family donated the journals to Acadia National Park. They recognized that he had a lot of information about the natural history of Oxbow, and they thought the National Park Service would be the ones who could appreciate that. And so around this time, College of the Atlantic started, and Acadia gave the journals to a professor at College of the Atlantic, who must have put them in the attic and forgotten about them until the turrets renovation. So when the turrets were renovated in the summer of 2013, those journals came back to the Park Service, and we digitized and scanned every page of those journals, and they're actually now available online. Anybody can go look them up. And the Quackenbush journals are about 5,000 pages. But one of the amazing things about Quackenbush was that at the end of his life, probably when he was no longer taking daily walks, he actually sat down and indexed everything in his daily journals. So he has an index of birds, and that's the bird folder, where he lists by species every observation that he had of a bird. Or he goes through by year and then goes, so for the spring of 1946, this plant leafed out on this day, this plant leafed out on this day. And so these incredible indices provide just a really wonderful data source. And he was so thorough. So here's the first page of his journal kind of blown up. And while his handwriting is not great, it's much better than Thoreau's. And so he writes a little bit about the climate. He talks about how February was feeling very spring-like. He heard a phoebe, and then he goes back and annotates it that it might actually have been a chickadee. And this data set is just really wonderful to work with. So we tried to find as much as we could about Quackenbush. And this is a photo from Oxbow. I went up there with Richard Premack, my advisor, in the hat. We worked with Jason Johnston, who has the binoculars. He's a professor at UMaine Presque Isle. And we actually found Quackenbush's house and barn. That's the barn behind us. And we tried to retrace his path walking through town to the post office, and looked at the plants that he was probably observing, saw some of the trees that may have been the same exact trees he was looking at in the 40s and 50s. And we tried to take that data set and create the same types of figures that we had done in Concord, Massachusetts. So in this figure, the x-axis is the mean April temperatures from cooler Aprils to warmer Aprils. And then the y-axis is the date from early May through mid-June. And the blue shows leaf out. So that's each data point there is 10 species, the 10 common tree species that he was consistently recording each spring from 1940 through 1955. And the gray is flower species. So these are the 15 common wildflower species that he was consistently recording from 1945 through 1957. And we can see the same trend that we saw in Concord, where in warmer springs, there's earlier leaf out and earlier flowering. And so we can quantify that shift. For leaf out, it's 2.5 days for each degree Celsius, and it's advancing just like in Concord. And for wildflowers, it's advancing 2.3 days for each degree Celsius. And again, it's the same direction that we saw in Concord, but a little bit less in magnitude. So Concord was advancing wildflowers by 3.2 days per degree Celsius. Quackenbush also had this really incredible data set of migratory bird arrivals, and he specifically had 9 species that he was recording over the course of 17 years. And incredibly, we were able to match this record with the ornithologist Bill Sheehan, who has been recording migratory bird arrivals for the same 9 species over a 17-year period from the 1990s through the 2011 was the last year we used. And so we were able to compare these 2 time periods. And what we found was that the birds were arriving earlier in the present time period than they had during Quackenbush's time. But they weren't correlated with spring temperatures. So it may not be a spring temperature in Maine that's driving this earlier migratory bird arrival, but perhaps something happening in their overwintering habitats that's cueing them to come up earlier. At the same time, I do want to point out that Quackenbush was pretty much just in Oxbow, Maine, walking around the same area, whereas Sheehan drives all over Aroostook County, which is a very large county, looking for first arrivals. So Sheehan might be finding earlier arrivals because he has a broader geographic scope. And so we have to consider the differences in methodologies that might be driving this. But it's still an important data set to look at because migratory bird arrivals have been studied in Maine. But in those studies, Aroostook County has been explicitly excluded. This is because historically there hasn't been enough data from northern Maine to include Aroostook County. So a number of papers that have come out from Wilson's lab at Colby have looked at historical observations of migratory bird arrivals, and they've never been able to include Aroostook County in that. And Quackenbush's data set allows us to take a look at this underrepresented county in the climate change literature. So again, this Hunting Guides journal contains really valuable climate change data, and the plants in Oxbow are responding to variability in spring temperatures, but the shift in days per degree Celsius is smaller than in Concord, Massachusetts. And our migratory birds are arriving earlier than in the past, but these differences might be an artifact of the methodology. All right, so just to wrap up, I wanted to talk a little bit about what I've been doing in Acadia National Park. I had such wonderful mentors and collaborators historically from Edward Rand and Annie Sawyer Downs and L.S. Quackenbush, and they've all inspired me to hopefully be a better field biologist and take much better notes during my time doing dissertation research at Acadia. So I just want to remind everyone of this figure where we have Thoreau's Concord, and each of those data points represents 32 species from a single year. So when you think about making a figure like this, it actually takes a lot of time. And so this is all of the data that went into that figure. On the x-axis, you have year, and on the y-axis, it's mean first flowering date. So this covers a lot of years and a lot of different observers from Thoreau through my advisor, Richard Premack. And when I started my dissertation, I just didn't think I had, 25 years to get that phenology data for my own work. And since Edward Rand and the Champlain Society were summering in Acadia National Park, or on Mount Desert Island before it was a national park, they weren't there for the spring phenology event, so they weren't recording leaf out and flowering. However, Mount Desert Island has this beautiful topographical variability. And so anybody who's been hiking in Maine knows that if you start at the trailhead in shorts and a t-shirt, by the time you get to the summit, you've added a couple layers, like a long sleeve t-shirt or a fleece or a hat, because the microclimate at the summit is different from the microclimate at the base of the mountain. So I used that to my advantage so I could collect a lot of data in a single year. And I was looking at Cadillac Mountain, Pematic Mountain, and Sargent Mountain on Mount Desert Island. And I took each of those mountains and divided them into different zones and looked at leaf out and flowering data across different zones and across the north and the south aspect of each peak. So I've had 7 elevation zones on each of these 3 mountains, and I monitored them twice a week in April, May, and June over 4 years during my PhD. And I was looking at 30 species, but I'm going to present some data from the 9 most common species. So species like lowbush blueberry, you can find across the entire elevation gradient, but some species are much more restricted. So I was doing this analysis on the most common species because we have the most data from them. So spring temperatures across these microclimates turned out to be a little bit more complex than we thought. We assumed the summits would be the coldest spots, and that was mostly true. And so I'm showing data here from Cadillac in 2016. This is the March and April temperatures. But the other cold spots on the mountain were not necessarily where you would expect. In fact, the lowest elevation site on the south aspect of Cadillac is pretty chilly, and that's because for the first mile or so of that Cadillac South Ridge Trail, you're in a dense conifer forest. So it's pretty cool underneath that canopy. And then the northern aspect at the lowest elevation, and even at a pretty high elevation, site 3 on Cadillac, is very open and rocky and receives a lot more sunlight than the closed canopy down below on the south side. So It led to some interesting discoveries about where the hot spots on the mountains are. And we tried to recreate that figure from Thoreau's time, or from Thoreau's Concord. And so on the y-axis, again, we have the date of leaf out from mid-April through mid-June. And the x-axis is mean spring temperature from cooler springs to warmer springs. Each color in this graphic is a different species. And each data point represents the date of leaf out for that species in a microclimate, and the microclimate matches up with the mean spring temperature. So each mountain is represented multiple times. Each elevation zone also, since I was there for three years with temperature data. And for 6 out of the 9 common species, we had a significant relationship between mean spring temperature and date of leaf out. And the 6 species are the ones shown at the top. So huckleberry, sheep's laurel, Canada mayflower, three-tooth cinquefoil, lowbush blueberry, and wild raisin. And when we looked at species that we had observed in Concord, Massachusetts, and on Mount Desert Island for leaf out, we found this pattern where across the board, they're all advancing, except for chokeberry, which is not significant. They're all advancing their leaf out in response to warmer springs, but the magnitude of that advance is not as strong on Mount Desert Island, very similar to what we found in Oxbow with Quackenbush's data. And this is true not just for leaf out, but also for flowering. We see the same pattern. And I set this project up on hiking trails. So my transects actually followed the North and the South Ridge hiking trail of Cadillac, and the same for Pematic and Sargent, because I loved doing this research, but I knew that at some point I would graduate, and I thought that it would be important for Acadia National Park to continue this type of work, and that if we had a fun project on hiking trails, we could recruit citizen scientists to continue this work. So one of the last things I did as a PhD student was to analyze different subsets of my data to kind of provide recommendations to the Park Service for different monitoring strategies. So I looked at lowbush blueberry data, the most common plant that we had. And then I looked at what would happen if I only had data from Cadillac. Would I still be able to see the same patterns? Would I have enough statistical power to make decisions? And what if I had data from the other 2 mountains, but not Cadillac? So I tried different combinations of ridges to see if we should encourage citizen scientists to hike a specific mountain, or if we needed all 3 mountains. And it turns out Cadillac on its own does a pretty good job of providing solid information about the phenology of these plants. And then I looked at, well, do you need to hike both the North Ridge and the South Ridge, or could you just send hikers up the South Ridge Trail? And it turned out the South Ridge is a really good trail to capture lots of variation in microclimate and phenology. And I looked at just low elevations and just high elevations. Oops, sorry. And I looked at what would happen if I reduced the number of years that I had data on. And reducing years is a terrible idea. We need to continue and add years. And so I ended up with these recommendations to include focusing on Cadillac southern aspects and more years for monitoring strategies with citizen scientists. But we found in general that this leaf out and flowering phenology is responsive to temperature across environmental gradients. And as in Oxbow, the shifts in days per degree Celsius is smaller than in Concord, Massachusetts. So to jump back to the broad conclusions from this dissertation, we have a changing flora on Mount Desert Island with lots of new non-natives. These floristic change patterns at the species level are different from in southern New England, specifically Concord. Records from the Hunting Guide, that's Quackenbush in Oxbow in the 1950s, and Ridge Transects, that's me, on Mount Desert Island in the 2010s, demonstrate advancing plant phenology in response to spring temperatures in Maine. But these phenological responses match the direction, but not the magnitude recorded in southern New England. And one of the great things about working on this dissertation in partnership with Acadia was providing these applications at the end. So we can say that we have this great example of the utility of historical records. We can highlight Northern Maine research in climate change, or Northern Maine as a region in research and climate change. We can provide a lot of local data from the historic data from Edward Rand and from my own ridge work for local management decisions. And we can support the climate change vulnerability assessments that Acadia has been working on. And hopefully we can initiate this citizen science project monitoring on hiking trails So hopefully this will be coming soon to Signs of the Seasons as I continue to work with Acadia to get this together. So I'd just like to thank all of the funders that have supported this research and my lab, especially Abe Miller-Rushing at Acadia National Park for mentoring this research. And thank you so much to Signs of the Seasons for inviting me to speak with you all. So with that, I'll take any questions.
That was awesome, Caitlin. Just much better than I even dreamed. So your research is so important to Signs of the Seasons and as many examples and future collaboration. I had a question. I see a couple of, I do see some questions. Well, I'll ask mine first. Were you looking at fall plant phenology at all? And the reason I ask is because, we are, of our different seasons, fall and winter are the most warming for Maine. I mean, very, very, very significantly more than spring and summer.
Yeah, so I personally haven't, but in our lab, Amanda Gallinat, who is about to finish up right now, wrote a really wonderful paper on autumn as the neglected season. Mm-hmm. And she has been working with Skudik, putting together the methodology to do studies on the timing of fruiting in non-native plants and native plants and how that's related to the timing of fall migration for birds leaving the area. So they've been working with bird banding stations and collecting data on when different plant species are producing berries, when those berries are ripening, and then looking at the bird poop basically in bird banding stations to see what seeds are in bird poop at different times of the fall, and trying to connect the dots through what's happening with autumn phenology. So it's an exciting area of research that I haven't done very much on, but I have helped Amanda pick apart some bird poop, and it's been a cool project.
That's great. Yeah, I have that paper. I should put that on our website. It's a great paper. So here's a question for you, and I realize that we're over time, but we started really late. So if people would need to leave, go ahead, but hang in there. If you have questions, type them into the chat box. If there are any unanswered questions, Caitlin said she's happy to answer those, and we can post those on our website along with the recording of this. So here's another question. What would you say is the most significant climate and/or phenology change that your research has uncovered, and why is it so significant?
So I think it's pretty interesting that what I found in Maine has been to match the direction of what we found in Concord, Massachusetts, but not the magnitude. And it's interesting because we're not sure why. And so one case might be that Concord, Massachusetts, it's right outside Boston. There's an urban heat island effect. And so warming not just from climate change, but also from being so close to a city. And so Concord might have just experienced a lot more warming. But we also might just have this artifact where Concord, we have so much data from Thoreau and Hosmer, who was a botanist in the early 1900s. And in Acadia, and even in Oxville, we're kind of just stuck in the 20th or 21st century. And so we don't have that whole range of differences. And so it might be a methodology thing. It might be there's actually differences in how different populations are adapting to differences in spring temperatures. I think it's probably very significant in terms of understanding what's happening across a region where, as I said, in Concord, Massachusetts, there's 300 plant species that are also in Acadia National Park. The plant communities are very similar, but they're responding in different ways. So trying to understand what's happening at that local level, it's maybe not a significant finding yet. It's more of a like, that's really interesting. We should look at that in a lot more depth. And I didn't expect it. I really thought that I would get up to Acadia National Park and there would be somebody like Thoreau who had that historic data, and I'd just like type it into an Excel spreadsheet and be like, oh, look, it's the same as Concord.
So that's good information. The other question I had is of course, that's a really special ecological habitat in
Mhm.
Mount Desert Island. And so we, a lot of our volunteers are working in their backyards and community places and don't have maybe those species. But we're definitely going to be adding some species because there are Signs of the Seasons observers that are out there looking at different species than the 19 that are on our list. And one of them is that we're thinking of that you have on yours is low bush blueberries. So
Um,
do you think of that as a good addition as an indicator species of how things are changing?
I do, and so one thing I didn't show you guys is I did an experiment on Cadillac Mountain with transplanting low bush blueberry from different elevations. So I had a summit population of low bush blueberry that I moved to mid and low elevations and vice versa, because we're really interested in species that can occur along these whole gradients. So you can find low bush blueberry pretty much everywhere in the park. And we've noticed anecdotally that the low bush blueberry on the summit is flowering and leafing out later than the low bush blueberry at lower elevations. And that makes sense. The summit is colder. So it might just be responding to that local microclimate. But if there's some kind of local adaptation also going on, where maybe the summit is more prone to late frost, or the summit warms up differently than low elevations, we'd want to know if those populations have evolved to have some kind of different adaptation than the same species at a different elevation or in a different microclimate. And that's important for understanding how species will respond to climate change across much less compressed gradients. So if you're looking at low bush blueberry from Massachusetts up to Maine, that's a similar kind of difference in microclimates. So low bush blueberry, you think of it as being very common, but trying to understand what's happening to it in different locations, I think, is really important. And it's also just a delicious study species.
Yeah. Well, thank you. That's great. So there are a couple of questions here. How is air pollution looked at in your study?
We didn't directly measure air pollution, but I think that could be an interesting thing to compare between Concord, Massachusetts and Mount Desert Island for sure. And it's definitely something that we could try to come up with a metric for when we're comparing all of the floristic change studies across New England. One of the things that was really interesting when I was reading all of those floristic change studies was that they eventually seemed to be pretty aware of each other, like they would cite each other as they came out, but it was often just so locally focused on, well, here's this one place, and here's what it looked like in 1800 or 1899, and here's what it looked like when we resurveyed it in 2005, and like there's a new parking lot, so maybe that's what happened. And so it was very focused on what was happening at the local level. Mm-hmm. But looking at something like air pollution gradients across New England might be a really interesting way to bring in that regional trend. I was trying to think of it in terms of conservation statuses or how urban to rural the gradient was, but I think air pollution could be another interesting factor there.
Definitely. That's a great idea. Okay, so here's another question. Could the cooling effect of the ocean be influencing the smaller temperature changes in Maine compared with Massachusetts?
Absolutely. Concord is inland, and so it doesn't have that maritime effect. It's not as tempered by being on the coast, and that might be one thing that kind of buffers climate change for Acadia National Park and all of coastal Maine. Yeah. So it could end up having coastal Maine being kind of this refuge in the future as the climate changes more dramatically inland.
Mm-hmm. Yeah, huge differences there. So another question is, what do you think is driving the dramatic loss of historically present species and the addition of new, not invasive species?
So I wish that we had kind of a middle or 1920s-ish flora for Mount Desert Island, because I think the creation of the auto road and the bridge onto Mount Desert Island probably had a pretty large impact. Mm-hmm. Around the time that Edward Rand and the Champlain Society were studying the island, it was becoming a summering place, but it wasn't attracting just the hordes of people that would eventually come up. It was still a pretty rustic location. And so I imagine that increasing auto traveling and increasing just the number of people probably brought in a lot of those new non-native species, but it also probably just led to habitat destruction or inadvertently people overpicking things like ferns. And so there's probably like that first jump of Mount Desert Island becomes very popular and there's more cars. And so we have an increase in non-natives and a drop in natives. But then I do think that the most recent climate change is probably driving some of that species loss. And when we look at places where there are multiple surveys of the whole flora, there's a really significant drop off, especially in declines in abundance and in local extirpations in the most recent resurveys. So places like Concord, Massachusetts, they have this, they have the Thoreau data, they have Alfred Hosmer in the early 20th century, and then another botanist, Ray Angelo, has been working in Concord, and he published a flora, I think in around 1970. And then when Abe and Richard went in and did their work around 2007, 2008, there's a really significant drop off from the 1970s to 2007, 2008. And it wasn't as dramatic in the previous time period. So there's something happening recently. And there have been a lot of papers that have tried to rule out things like white-tailed deer, tried to look at specific events like habitat destruction. And our work in Acadia, we try to look at habitat loss or look at species loss across different habitats to try to identify if a specific habitat, for example, grasslands, which are becoming much more wooded, especially since it's a national park and no one is out there actively farming or cutting grass. But every habitat category is losing species, which indicates there's something more region-wide or more global happening. And it's really hard to find like the direct evidence or the smoking gun of like climate change impacted that plant species for sure.
Yeah. Well, I don't see any other questions, but if any do come up after the fact, then
Sally just put up something about oxbow.
Okay.
Oh, did I see something similar in oxbow? We actually don't know very much about the abundance of plants in oxbow, so it's hard to determine how the plant abundance has changed in oxbow. We just have our data from the 40s and 50s on their leaf out and flowering times. And we've actually been working with Jason Johnston and his colleagues at UMaine Presque Isle, and they've been monitoring the same species both in Presque Isle and in oxbow. So we have some updates on how those species have responded to recent years. But unfortunately, we started this project after 2012, so we're missing that really great outlier year of the extremely warm spring. But hopefully we'll have something to talk about with oxbow pretty soon, and we're hoping to publish that paper this fall.
Great. I'm looking forward to that. Actually, I had a quick question. Were there no sites in common with Sheehan and Quackenbush as far as the bird phenology goes?
No, it was mostly just that so oxbow is kind of in the middle of nowhere. So Sheehan hadn't been out there, I think, until we started talking about it. But just that he was traveling so widely across the county that he was just covering a much larger geographic range.
Right.
It would be slightly more comparable if he had just been like in Presque Isle and hanging out around Presque Isle itself, but he needed to travel across the county, which is very common for birders. I totally understand wanting to see the first season.
Right. Okay. Any other questions before we close this out? Well, I can't thank you enough, Caitlin, for your presentation and great answers to our questions and increasing our knowledge on this. This is wonderful, and look forward to, your other work and that paper and working together.
Great, thank you so much. It's been really wonderful working with Signs of the Seasons, and it's awesome to be able to talk about my work. And as I said before, I had a good time doing my defense, or as much of a good time as you can defending your PhD, but it's really nice to get to talk about the human side of the research a little bit more.
Well, it's very, very interesting, and I think that getting some comments about that as well. So that's we really appreciate it. Thanks so much. Lots of
Thank
applause. Thank you all for participating.